Chapter 4, Section 16, Oxygen and Microbial Growth. We're a bit biased. We mammals, we seem to think everything needs to have oxygen, just like we do. No. Yeah, we're aerobic. We need oxygen, about 21%. We need to respire. We need to breathe in O2, breathe out CO2. But a lot of the microbes, most of the microbes out there are not going to be true aerobes. There's going to be some that are microaerophilic. They can use oxygen as their final electron acceptor. They need it, but they need limited amounts of it. Much, much lower than what we're doing. And then there's facultative aerobes. You know what? If it's there, we'll use it. If not, we'll use something else. We talked about this last chapter with E. coli and its final electron acceptor. Oxygen, if it's there, and if it's not, well, it'll use nitrate, as its final electron acceptor. So you go and you look. There's aerobes, anaerobes, obligate, facultative, microaerophilic. The anaerobes, there's the obligate, keep all oxygen away. Oxygen is harmful, lethal. Keep it away, do not want it. And then there's the aerotolerant. They don't want it. They grow better when it's not there, but if it's there, well, all right, whatever, we'll still get by. We're just not using it. The microaerophilic, low levels, the levels get too high, it becomes lethal. Facultative, not required, but you know what? We can use it. There's our friend E. coli. So the anaerobes, they don't need oxygen. They have a different final electron acceptor, or they just do fermentation and they don't even bother with the whole, electron transport and whatever. Aerotolerant, and then the obligate anaerobes. Aerotolerant, don't want it, but if it's there, we're just going to ignore it. The obligate, keep it away. Keep every bit of it away. Cannot handle it, cannot grow with it. It will kill us. So you have to keep this in mind when it comes to anaerobes. The aerobes, hey, you know what? They're easy. Put them in a broth or whatever. Put it in the incubator or a shaker incubator. Just keep them shaking. Keep them moving. Keep that broth liquid moving. And it just keeps aerating it. Boom, they're happy. The anaerobes are going to be different. Okay, the anaerobes, well, first you're going to have some sort of a reducing agent there to remove any and all oxygen from the liquid broth. Because just the sheer, the sheer act of you pouring water aerates that water, increases the concentration of oxygen held in that water, in that liquid. Stirring it, moving stuff around, sloshing it, causes it to aerate, get more oxygen in it. So you have to use these reducing agents like thioglycolate. Thioglycolate will go through and it will bind up any gaseous, any molecular oxygen present in that liquid. And it will hold it, sequester it away, keep it away from the microbes. You can use certain dyes like resazerin. Tells you whether or not you have a high or a low oxygen concentration based on color changes. You can put everything that you've treated with reducing agents, removed all the oxygen as much as you can, used vacuum pumps to pull the gases out of the liquids. You can do all this, and then you can use what's referred to as a glove bag. It's an environment, a sealed environment that all the oxygen has been displaced by either carbon dioxide or gaseous nitrogen. And what's interesting, we can go through and we can, you know, grow microbes in test tubes, and we can get an indication of aerobe, anaerobe, facultative, based on their growth characteristics. You take tubes with a liquid broth. You put different bacteria in them and not shaking or anything. You just put them in there and you let them grow. If the microbes grow up here at the surface, this is the oxic zone. This is the highest oxygen concentration. That means these are aerobic. If they grow high there and then spread out a bit, well, then they're, facultative aerobes. They can grow in the presence, they can grow in the absence just as well. They grow just below the surface, like they're showing here. Not quite the oxic, more into the anoxic, but still microaerophilic. Down here at the butt, well, that's going to be the lowest to almost no concentration. It means these are going to be anaerobic. The oxygen that's up here at the surface is damaging to them. So they need to be away from it. Looking at my notes here. So that's how we can sit there and tell. Aerobe, anaerobe, microaerophilic, facultative aerobes, and facultative anaerobes, whatever. I mentioned a glove bag. This is a glove bag. This is a portable workstation. It's a sealed environment, sealed by this plastic. There are pumps that you can't see here that will draw out the atmosphere, replace it with some other gas. Carbon dioxide, nitrogen, so that this is now free of oxygen. So you can work with your microbes here. This back here is, your incubator. So you can store at certain temperatures your cultures. Different pumps. You see here one, there's one that will remove the atmosphere, replace it with something else. If you need to move stuff in and out, that's what this chamber right here is for. You open this little chamber, you put the stuff in there, you close, seal the chamber, pump out the atmosphere, replace it with something else, put your hand in here in these gloves, take out whatever. It's now inside. You need to remove it, you do the reverse. This is if you're working with a true anaerobe. Okay, this is something that it cannot be in the presence of oxygen because it will be damaging if lethal. If it's something that's facultative, or you can, is slightly aerotolerant or whatever, that's what these canisters are for with the gas pack. You do whichever you're going to do out there on the table like you do in lab. You put your plates or your broth test tubes into this canister. Seal the canister with one of these gas packs. The gas pack here has a chemical reaction, twofold chemical reaction. One is a series of crystals that are going to absorb any oxygen from the environment within the canister, and the other is it's going to produce carbon dioxide, which is going to fill in where the oxygen is being removed, so that this way this maintains an internal temperature or an internal pressure. A lot of times people are sitting there looking at me when I sit there and say oxygen is deadly. Oxygen can be deadly. And they're like, no, we breathe it. It can't be that bad. And it's like, yeah, it actually can. You take O2. O2 that you breathe in is not that bad. But O2 can be broken down, or it can combine with other things in your cells that, superoxide anions. It gains an electron, O2 negative. Hydrogen peroxide can break down to become hydroxyl radicals and or superoxide anions. O2 gains an electron, strips it off of something, because remember I keep saying that in lecture. O2 is a greedy bugger. It will strip electrons. You get superoxide. O2 plus that, and it picks up some hydrogens that it stripped off someplace. You now have hydrogen peroxide. Hydrogen peroxide can break down into water and a radical, hydroxyl radical, which is just as damaging as the superoxide anion. Now, thankfully, we have enzymes that will go through and will counter all of these. Most aerobes have evolved to have mechanisms for countering oxygen radicals. Catalase, peroxidase, takes hydrogen peroxide and turns it into oxygen and water. Skips the whole hydroxyl thing. Superoxide dismutase takes our oxide anions, binds it to hydrogen peroxide, reforms this to make, takes this with water, makes hydrogen peroxide in just plain old O2 bubbles. Superoxide reductase. You're going to see these in strict anaerobes. Not all anaerobes have these abilities. These are one of the things we can sit there and test for in lab. If by chance that strict anaerobe gets exposed to oxygen, it can take the oxygen radicals, convert them to peroxide without producing gaseous oxygen. The hydrogen peroxide will then just be excreted out of the cell. So catalase takes hydrogen peroxide, turns it into water and O2. Remember when you were a little kid, you got a cut or something and mommy poured the hydrogen peroxide from the brown bottle on the cut and it bubbled? Yeah, that's the cells in your tissues and some of the bacteria in your cut that have catalase. Turns out all of our cells have catalase. So it's taking the hydrogen peroxide, breaking down to water, and there's what's the bubbles. Peroxidase takes the hydrogen peroxide, NADH, and hydrogen, and will oxidize. Peroxidase will oxidize NADH to make water and oxidize NAD. It reduces hydrogen peroxide to make two waters in NAD. Superoxide dismutase takes our radicals here, our oxides, and hydrogen makes peroxide, which then catalase can then turn into water and oxygen, more gaseous oxygen. So when you look at that combined, O2 negative plus hydrogens make water and a whole bunch of bubbles. See, it's one of the tests you're going to do in lab. You take a drop of hydrogen peroxide, and then you take a loop full of your bacteria that you're trying to characterize, you're trying to test for catalase capabilities. Does it have the enzyme catalase in it? Because not all of them do. You take a loop full of your bacteria, mix it in with a drop of hydrogen peroxide, and then you look for this. Bubbles.